Dilation is important question, and also the venous drainage, mainly the coronary sinus is very important. Okay, so venous drainage, the venous blood of the heart is drained by these three types of veins: coronary sinus, anterior cardiac veins, and veni cordis minimi. Okay, see here, this is the position of the coronary sinus in the atrioventricular groove, in the posterior part of the atrioventricular groove. About 60% of the venous blood from the heart enters into the right atrium, whereas remaining 40% enters into the different chambers of the heart. Okay. Okay, next we will discuss these three types of veins. First we will discuss coronary sinus. Coronary sinus is important question, comes as a short note. Okay, so it is a major vein of the heart. See here, this is coronary sinus. Size is about 2 to 3 centimeter long. Its location, it is located in the posterior part of the atrioventricular groove here. How it develops? Development you will understand during the cardiovascular system development. Okay, you just know the terms. It develops from the left horn of the sinus venosus and a part of the left common cardinal vein. So from these structures the coronary sinus develops. Now beginning of this coronary sinus, it begins as the continuation of great cardiac vein in the left part of the atrioventricular groove. See here, this is great cardiac vein. Here it has been pointed. Great cardiac vein lies in the anterior interventricular groove, in between the two ventricles on the anterior aspect. Then it turns round around the left border of the heart, and from its termination the coronary sinus is started. Okay, so it begins as the continuation of great cardiac vein in the left part of the atrioventricular groove. This is the beginning of coronary sinus. Now termination. Coronary sinus opens in the right atrium. Here this is the opening of the coronary sinus, and its opening lies in between the opening of inferior vena cava and right atrioventricular orifice. This position is important. Opening lies in between the opening of inferior vena cava and right atrioventricular orifice. And the opening of coronary sinus is guarded by non-functioning rudimentary Thebesian valve. This Thebesian valve, it is important for your viva, and also during viscera it is asked. Opening of coronary sinus, it is guarded by a non-functioning rudimentary Thebesian valve. Okay. See, this is view from the posterior aspect. So this is posterior part of the atrioventricular groove, and this is the position of the coronary sinus. These are the ventricles. This is left ventricle. Here is the right ventricle. Okay. These are the atrium. Left atrium, right atrium. Here the superior and inferior vena cava draining into the right atrium, and 4 pulmonary veins are draining into the left atrium. These are the two divisions of the pulmonary trunk. Here is the arch of aorta. Okay, and see, these veins are draining into the coronary sinus. This is inferior or posterior vein of the left ventricle. On this side, left ventricle is lying, draining the wall of the left ventricle. This is middle cardiac vein. Middle cardiac vein lies in the posterior interventricular groove, and it is tributary of this coronary sinus. And this is small cardiac vein. Small cardiac vein on the right part of the posterior interventricular groove. It is also a tributary of the coronary sinus. And see here, this small cardiac vein originates as the right marginal vein. This is the right marginal vein lying along the right margin of the heart. Okay, and here is the left marginal vein, which is lying along the left border of the heart and draining into this coronary sinus. Okay, and the coronary sinus starts as continuation of this great cardiac vein. Okay, and this is one oblique vein of the left atrium that is also a tributary of the coronary sinus. So coronary sinus receives these tributaries. All these tributaries we will discuss in details. First is great cardiac vein. It begins at the apex of the heart. Here is the apex of the heart. You will know during the viscera classes different parts. Here is the apex, and posteriorly base is formed by the two atrium, mainly the left atrium. So in the great cardiac vein, it begins at the apex of the heart and it runs upward in the anterior interventricular groove, and then it runs in the left part of the atrioventricular groove, here left part of the atrioventricular groove, and turn around the left heart border and opens in the coronary sinus. So this is the course of the great cardiac vein, and it receives the blood from the left marginal vein. Here this left marginal vein is draining into the great cardiac vein. Next tributary is middle cardiac vein. It begins at the apex of the heart and runs in the posterior interventricular groove. Here this one is the middle cardiac vein, runs in the posterior interventricular groove, and it opens in the right end of the coronary sinus. Here it is opening into coronary sinus. Okay, next is small cardiac vein. It lies in the right part of the atrioventricular groove. Here is the small cardiac vein, lies in the right part of the atrioventricular groove, both on the anterior aspect and also in the posterior aspect, and opens in the right end of the coronary sinus. Here it is opening into coronary sinus, and it receives blood from right marginal vein. You can see this right marginal vein is draining into this small cardiac vein. Okay. Next is posterior vein of left ventricle. Also it is known as inferior vein of left ventricle. Here it is lying, inferior vein of left ventricle or posterior vein of left ventricle. It drains the diaphragmatic surface of left ventricle and it opens into the coronary sinus just left to the middle cardiac vein. Here it is lying left to this middle cardiac vein, draining the diaphragmatic surface of left ventricle. Now next vein is the oblique vein of left atrium or vein of Marshall. Here is the oblique vein of left atrium or vein of Marshall. It is a small vein that drains the posterior surface of left atrium. Here it is coming from above, here atrium is lying. So it is draining the posterior surface of the left atrium, and it opens into the left end of the coronary sinus. See here opening at the left end of the coronary sinus, and development, it is remnant of left common cardinal vein or duct of Cuvier. Okay. This development of important because it comes as a MCQ question. Okay. Remnant of left common cardinal vein or duct of Cuvier. Next is right marginal vein. It runs along the inferior heart border. Here is the right marginal vein. Inferior heart border, it extends from this apex of the heart up to the opening of the inferior vena cava. This is the inferior border. Here is the left border along which this left marginal vein is lying. And here is the right border. Right border is formed by joining of these two great veins, superior vena cava above and inferior vena cava above. So you can see the right marginal vein is lying along the inferior border. Okay. It runs along the inferior heart border and opens into small cardiac vein. Here it is opening into small cardiac vein, and sometimes it opens directly into the right atrium. Okay. That is some variation of opening. Now left marginal vein, it runs along the left heart border and drains into coronary sinus. Here is the left marginal vein. It runs along the left heart border and draining into this great cardiac vein and drains into coronary sinus. Okay, so these are the three veins draining the heart. Anterior cardiac vein, which opens directly into the right atrium, coronary sinus, and the veni cordis minimi. They open in all the four chambers of the heart, most numerous in the interatrial septum. So, coronary sinus is the main vein of the heart, lies in the atrioventricular groove on the posterior aspect, and termination it opens into the right atrium and guarded by a valve, Thebesian valve. Okay. And these are the tributaries of the coronary sinus: great cardiac vein, middle cardiac vein, small cardiac vein, posterior vein of left ventricle, oblique vein of left atrium, left marginal vein, right marginal vein, which opens in small cardiac vein. Okay, so these are the tributaries of the coronary sinus. Okay, so if coronary sinus comes as a short note, you have to write down it beginning, then termination, guarded by a valve, and all these tributaries you have to write down. Okay. Next is anterior cardiac vein. They are 3 to 4 in number. See here, this is the position of anterior cardiac vein. 3 to 4 in number. They are small veins that drains the infundibulum of the right ventricle. Infundibulum is the smooth part of the right ventricle. Okay, and they open directly into the right atrium. See here, they are opening into the right atrium. Here is the right atrium, draining directly into right atrium, and they are 3 to 4 in number. Next is veni cordis minimi or Thebesian valve, Thebesian veins. Okay, they are small veins that opens into all the four chambers of the heart through foramina minimarum. Okay, or foramina venerum minimarum. Here, see this is right atrium has been cut open. Okay, to see the interatrial septum. Here, this is the rough part of the right atrium. These muscular ridges are known as musculi pectinati. Okay, and here is the crista terminalis, which is a longitudinal smooth ridge. Okay, along the crista terminalis, here along the right border of the heart, it has been cut open to show the interatrial septum. And these small openings in the interatrial septum, you can see these are the positions of the foramina venerum minimarum. Okay, and these are most numerous in this interatrial septum. These openings are present in all the four chambers of the heart. Okay, so these are small veins open into all the four chambers of the heart through foramina venerum minimarum. Okay. This is enlarged view of this interatrial septum. Interatrial septum, here these are the openings of foramina venerum minimarum. Here is the opening of inferior vena cava. Okay, and in this part, here is the tricuspid annulus or connection between the right atrium and right ventricle. Tricuspid valve is there. So you can see, and this is the opening of the coronary sinus. Okay, so you can see the position of opening of coronary sinus. It lies between the opening of inferior vena cava and in between the opening of tricuspid annulus or right atrioventricular annulus. Okay, so the opening is lying in between these two openings, opening of inferior vena cava and tricuspid valve. Okay. Okay, that's all. So this is the venous drainage of the heart. Coronary sinus, very important for your short note. And other veins are veni cordis minimi and anterior cardiac vein. Okay. Any queries? Okay, you have understood? Okay, see the position of different veins, tributaries. See, this is posterior view from the posterior aspect. Okay, position of these veins are important. During viscera of the heart, you will be asked which artery is lying in which groove and which vein is lying in which groove. Okay, so you can see these two are very very important. Middle cardiac vein, this is view from the posterior aspect. So this is posterior interventricular groove. So middle cardiac vein is lying in the posterior interventricular groove, and this part is the coronary sinus, about 2 to 3 centimeter in length, lying in the posterior part of the atrioventricular groove. Okay, and here in this right part, here lies the right part of the posterior atrioventricular groove. Here lies the small cardiac vein, and on this side, left part of the atrioventricular groove, here lies the great cardiac vein. And great cardiac vein also lies in the anterior interventricular groove. This is anterior view of the heart. Here is the position of great cardiac vein in the anterior interventricular groove. Okay, and here is opening of the coronary sinus into the right atrium, guarded by a valve known as Thevesian valve. Other tributaries you can see, here is the inferior vein of the left ventricle. This is right marginal vein along the right inferior border of the heart, and here is the left marginal vein along the left border of the heart. Okay. And in this view you can see the base of the heart. This part is the base of the heart, which is formed by the two atrium, right atrium and left atrium. Mainly 2/3 part is formed by this left atrium, 1/3 part is formed by the right atrium, and this is diaphragmatic surface of the heart. It is formed by the two ventricles. Okay. Here also this is the apex, here is the inferior border you can see, and this is left border, and here is the superior border. Okay, and this is the origin of two great vessels. Arch of aorta is originating from the left ventricle, and pulmonary trunk is originating from the right ventricle. Okay, and at the region of origin, they are guarded by valves with three cusps. Okay, pulmonary valve and aortic valve, and just below the valve there is the smooth part. Smooth part is lying. Okay. So any queries regarding this topic? Okay, different parts. You have to learn about the apex, base, which chamber is forming which presenting parts of the heart, different borders, surfaces of the heart. Okay, that will be shown during your demonstration class, viscera classes. Okay, no queries regarding the venous drainage? Okay, so that's all for today. Okay, thank you.
So, once again, with this embryology, general embryology, we have covered the difficult most part of this general embryology till now. So, this rough riding with this general embryology is over almost. So, what you have done so far in last 4 weeks or nearly 4 weeks about the embryological development, we learned this particular single cellular gamete after meeting with this other counterpart from this unicellular zygote that transforms into, divides into, and happens to be forming into different layers. So, from one cellular layer, it forms cavities, it forms different kinds of layer which is going to be transformed into different parts of the body. So, we learned that particular embryo initially was a two-layer space, we call them as a disc, germ disc, which was covered in above and below by some cavities which was lined by some cells. And eventually these particular two-layer cells, which was their germ disc, transformed into third layer, which was the most important part of this development. We call them as gastrulations, which have seen that migrations of the cell, we call them as epiblast cell. They are more versatile, or most versatile cells that ever human mankind had, which can transforms into any kind of cell. So that particular epiblast cell transforms into the mesenchymal cells, which is going to be the mesoderm cells, and then the mesoderm cells also been replaced by these endoderm cells. So we got the three layers, and each of these particular three layers we have learned that covered by this particular covering yolk sac and this particular amniotic cavity. And within this particular element, this epiblast layer, we see that particular the neural development, which is the formations of the primitive brain that start happening with this primitive streak, primitive pit, and that particular new relations is triggered by this infolding of this epiblast as well, which is going to give rise this new relation, the neural tube, which was the flat thing, and then it merged together, formed a fold, and eventually get rise to this tube formation. And that particular tube elongated, differentially grew in the cranial part. It has made this particular brain or brain vesicle, which is having a three-part, forebrain, mid part, and the hind part, which happened there. And then we have the spinal cord, which was growing at this particular point. Now, this particular region we have sailed through, and we learned this particular thing, which is going to talk about the easier part of this thing. So, before we go into this particular further cellular transformations of this layer, we just want to just talk about briefly about very briefly about this particular element. We call them as allantoids. I skipped that particular slide when I was talking in the previous day. Allantoids is a kind of tissue derivative that is contributed by this yolk sac. So, we understand the yolk sac, which was covering this developing embryonic disc at the bottom side, and that particular yolk sac has a projections like outpouch. That outpouch we call them as allantoids, and that particular allantoids makes a great sense in formations of the urinary bladder formation. So, when again we are going to talk about the individual organ development in the embryological developmental part, we will be coming again to talk about the allantoids. So, this is allantoids what we want to make. So, we have this particular germ disc in the central part, amniotic cavity, which filled up the fluid in the upper part. Lower part we call them as yolk sac or primary or secondary yolk sac. And this particular yolk sac, it has a connections towards this part, we call them as the connecting stalk. The connecting stalk, we know that this is going to become the umbilical cord in the future times. Now, this particular yolk sac, you see that there is a pouch which is extending towards this connecting stalk. So, this small pouch which is projecting, conical pouch, we call them as allantoids, which makes a lot of sense when we go talk about development of the urinary bladder. This tiny tissue of allantoids pouch transforms into urinary bladder or a portions of urinary bladder. So, again when you go talk about this urinary bladder or urinary system development, we will readdress this particular element, we call them as allantoids. Again we will be talking about this. Now, why we are talking about this particular element, allantoids? Because today what we are going to do today, we have to understand the position of allantoids in the embryonic disc, where this particular allantoids is present in the embryonic disc, and we are going to talk about something beyond this particular disc, which is going to make this particular shape of this embryo today. So, for the day, the task is we are going to talk about this particular three germ layers that we have talked: ectoderm, mesoderm, and the endoderm. Now, what we have to do, we have to learn that what individual component of this cellular layer gives rise to the human parts of the body. That is the thing we are going to talk. And the second here, we are going to talk about, we call them as folding of embryo. So far we have seen this particular disc is a flat structure, just like a leaf. Now we have to make a tube, just like we have formed a tube in a neural tube. So that particular cylindrical structure that we are born with, we have to develop that. So for developing that, we have to go through a process, we call them as folding of embryo. That's the two elements we are going to address today. So first thing we are going to talk about the derivatives of this element. So this is what we are going to make. So we have three layers. Each of these particular layers is required to happen because individually three layers going to give rise different organs. So for example, we talked about this ectoderm. We call them as epiblast tissue transforms into ectoderm. That ectoderm form the neural tube. We've seen that superficial layer becomes neural crest, which coming out of this particular ectodermal tissues, and then it transforms and migrates out, and this particular neural crest tissues forms this ectoderm. So individually each of this particular layer give rise to this particular element. Say for example, ectoderm gives rise to the central nervous system and this particular brain formation. It gives rise to the skin formation. It gives rise to formations of the adrenal gland medulla part. Compared to the mesoderm, so this particular mesoderm eventually give rise to the kidney, reproductive organs, which is coming, heart, blood vessels, etc., spleen, all these organs which is coming from the mesoderm. Endoderm, the GI tube, that means the intestinal tube, which we are going through this through the embryology classes. We are going to talk about the liver, we are going to talk about this bladder, urinary bladder, etc., etc. They are developed from this endoderm. So each of these particular component coming from each of these layer. That's what we have to know. So we call them as derivatives of germ layers, which we see that had already surfaced last year as a 14-mark question about embryological development. So this particular question previous year was surfaced there in the examination. So as an exam important area, it's not that important, but you have to know because it might be surfacing again with this particular short notes. So derivatives of the ectoderm, what develops from the ectoderm that you have to know. It develops from this particular central nervous system, it develops in the glandular system, it develops ear, it gives the hair follicle, sebaceous gland, mammary gland, pituitary gland, and the teeth, part of the teeth. So all these elements which is ectoderm is derived of. So we call them as ectoderm, going to give rise to the neuroectoderm and surface ectoderm. From the surface ectoderm, all these elements, epidermis, skin components which develops from it. So this is the derivative of the ectoderm that you have to keep in mind. It's easy in a sense. So central nervous system and skin, if you can remember this particular thing develops from ectoderm, we are good. Now the elaborate thing that we have to do right now is this, that we have to know a lot about the mesoderm because that's a complex thing, and we see these mesoderm derivatives, we see that coming as an exam questions. So this is the bilaminar germ disc, going to be triamlaminar formation of this primitive streak, primitive pit, and this buccopharyngeal membrane and this particular cloacal membrane which is forming there, and these ingressions of the cell is happening there, and this particular two layers soon becomes three layer. So we have acquired another layer, we call them as mesoderm. This mesoderm we call them as intra-embryonic mesoderm, that is placed between this hypoblast, epiblast, in between them it is placed. So this is how this particular element is produces. This red tissue which you are seeing here is the mesodermal tissues, and this particular mesoderm is lying within this embryonic disc. That's why we call them as intra-embryonic mesoderm. Now this particular intra-embryonic mesoderm goes on changes or transforms. That transformations we have to know. That's the part which you are going to address. So see that this is a sagittal section that you are seeing here, cross sections from it. It's taken out. This is a developing notochord which is placing over here, and this particular notochord is formed in this C picture, bottom one, and next to right side and left side we see the red layer which is this particular the intraembryonic mesoderm. The blue one is the ectoderm and the yellow one is the endoderm. So three layers we have them. So we will be focusing what happens to this particular red element, we call them as intraembryonic mesoderm. So this particular notochord stimulates this particular intraembryonic mesoderm to grow. It grows and it populates and makes a shape. So this particular red tissue that we are seeing here on the right side and left side of the notochord, they go in a bulk or go in thickness, and they transform into three different components. So if you see these transformations of the intraembryonic mesoderm, this is the same section that you are seeing here. You see that this is notochord, and this particular intraembryonic mesoderm, the red tissue, is getting in volumes from A to B. Now, this particular region which is close to this particular notochord, this intraembryonic mesoderms becomes heavily thick. So, this particular mesoderm which is around this particular notochord, we call them as paraxial mesoderm. So, by the axis which is formed by this notochord of this embryo, so by the right side and left side of this axis of the notochord, we give this name of the intraembryonic mesoderm as paraxial mesoderm. Next to that, most laterally, the part of this mesoderm, we see that there is an appearance of holes in here. So, this particular vacuum or vacuoles which is appearing on the lateral part of this intraembryonic mesoderm, we call them as lateral plate mesoderm, where this particular vacuum appears slowly, small hole appears, small cavity appears here. So, this part we call them as most lateral. If it is a midline one, this is a lateral side, easily can be figured out, and this region we call them as lateral plate mesoderm. Now, then we are left with the space which is lying between this midline or close to midline and the lateral part, intermediate region. The intermediate region of this mesoderm, we call them as intermediate mesoderm. So, there are three different domains of this particular mesoderm or intraembryonic mesoderm can be figured out. So, we have a better visual on this. So, this is the scanning microscope picture, and we have used three different color codes. So, this green one is the notochord. This is the neural tube which is formed by the induced by this guy, which is formed over there. Roughly, this picture is corresponds to the age of 4th week, progressed 4th to 5th week of age of this embryo is there. Now, you see that this particular region is the mesoderm. So, this region is mesoderm. This is the upper surface which is ectoderm, and this one is the endoderm. So, these are the three different layers which is there. Now, along this axis next to that, this mesoderm, we call them as the yellow one, we call them as paraxial mesoderm. Next to that, we have these lateral extremes, we call them as lateral plate mesoderm, which holds the cavity. You see that which has a vacuum in it. And in between them, we have a tiny area, we call them as intermediate mesoderm. So, these three transformation or three segmentations of mesoderm is important. You have to memorize these names, because memorizing these names, segmentations of these thing develops into different organs. We'll be talking about very soon. From each of these component develops three different regions of the body. That's what we are going to talk today. Now, you see that within this particular lateral plate mesoderm appears a coelom. We call them as intraembryonic coelom. So, this particular coelom was not there before. So, here this particular vacuum or cavity which appeared, they joined together, and after joining together they create a single cavity. That single cavity which is present in here, we call them as intraembryonic coelom. Now, this particular cavity which happened in the lateral plate mesoderm, intraembryonic coelom, splits this mesoderm into two layers. One is the layer which is going along with this layer which is the amniotic layer, and which is dropping down over here with this yolk sac layer. So, we get these two layers of this lateral plate mesoderm which is holding a cavity in between. This cavity we call them as intraembryonic coelom, that future going to be the intraembryonic body cavity. The intraembryonic body cavity, that means the thoracic cavity, the abdominal cavity together. Without partition, this was happening. Getting it what I am trying to say? So, this transformations which is taking place over here is this coelom, we call them as intraembryonic coelom, which in future going to be intraembryonic body cavity, that cavity which is occupied by the lungs, livers, etc., this cavity which was yet to be developed. And then we have this layer which is the mesodermal layer. So, the mesodermal layer of this intraembryonic mesoderm which is covering this yolk sac, we call them as visceral layer of mesoderm, and whereas this covering of this lateral plate mesoderm which is covering around this particular amnion, we call them as parietal layer of mesoderm. So, we get the same parietal layer and the visceral layer, splankling layer and somatic layer of mesoderm which is around this particular coelom or cavity. So, once again we get this particular intraembryonic mesoderm which eventually segments into three different domains. That three domains we call them as this one next to this particular midline, we call them as next to this midline প্যারাক্সিয়াল মেসোডার্ম। ওই 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 দুজন উঠে চলায়, এখানে চলায়, ওই পুরো বেঞ্চটাই চলায়, ওই যারা গাদাগাদি করে বসে আছে, অল অফ ইউ, তুই থাক এইখান থেকে, হ্যাঁ চলায় পুরোটা, চলায় এখানে চলায়, মুভ ওভার হিয়ার। So we have left with 3 mesoderm which is transformed over here, which is paraxial mesoderm, intermediate plate mesoderm and lateral plate mesoderm. Within this lateral plate mesoderm we have the cavity, intraembryonic body cavity, which splits into 2 layers. We call them as somatic layer or parietal layer of mesoderm and splanchnic layer, visceral layer. A layer which continues with this particular yolk sac we call them as splanchnic layer, and the layer which continues with this particular amnion we call them as somatic layer or parietal layer of the lateral plate mesoderm. What do? They hold the cavity which is going to be the intraembryonic coelom. That particular coelom is going to be the future body cavity. So this is what we are going to talk. Now this particular region we call them as the paraxial mesoderm, the yellow one, shows further transformations. Soon we see that this particular layer which is known as this paraxial mesoderm go on segmentations like this. So this particular paraxial mesoderms make small cubical blocks like this, one after another, and this particular cubical blocks do not appear at the same point of time. They appear slowly. They appear from this particular neck region, we call them as occipital region, and then they gradually keep on forming and form down. So this cubical block we call them as somite or somitomere. So these names you have to keep in mind. So we call these cubical blocks of this paraxial mesoderm. So not other mesoderm like intermediate mesoderms, so the lateral plate mesoderm, they do not show this particular somite formation. Only this particular yellow guy which is shown there, that particular region we call them as paraxial mesoderm, shows on further development, keeps on segmenting, breaking down into segments. So these segments they keep on forming starting from this 20th day of the development, they keep on forming there, which develops in the occipital region first, and then slowly by the end of the 5th week the total count of this somite or somitomere is 44. Now they keep appearing every week or every day, they keep appearing, the number increases every day. So if you count this particular number of a developing embryo, we can get an age of this embryo. So this particular appearance of the somite can be measured, can be recorded, and this things appears as a particular time. We call them a somito clock. So this particular observing the somites can exact date of this embryo can be figured out. That's a separate entity. So this particular somite which is formed around this particular paraxial mesoderm surrounding this particular developing neural tube, you see that this is neural tube which is folding over here in the cervical region. It folds first and then zips up and zips down, and then we see that this is 2 pores which is eventually present, anterior neuropore, posterior neuropore. They closes eventually 4th week of age and 5th week of age, or else all the neural material will be washed out. We see that clinical complications if they are failed to fuse, what happens to the individual. But anyways, let's focus on this particular region we call them as paraxial mesoderm. So paraxial mesoderm breaks down into region we call them as somite. So this is the same picture that you are looking at, little bit advanced stage. This is the developing neural tube, this is the notochord that you are seeing here. This region is paraxial mesoderm which actually form the somites in this region. So this particular somite which is formed in cubical block can be split into 2 parts. So this particular somite can be split into lateral half and the medial half. The lateral half we call them as dermatomyotome, and the medial one we call them as sclerotome. This particular names we have to keep in mind, so which is the future development of this somite. So from the sclerotome it develops the scleral tissues, and dermatomyotome, if you split this particular name dermatomyotome, it gets the dermis and a myotome. That means from this particular dermatomyotome component of this paraxial mesoderm of the somite develops derma, that means skin, and muscles, myo. So the skin and this particular muscles develops from this lateral component of the somite which is paraxial mesoderm. The medial component forms the sclerotome. Now we'll focus on the sclerotum. What happens to the sclerotum? This notochord gives an inducing signal and pulls this particular sclerotum tissues. Now this particular notochord and the neural tube on top pulls this particular sclerotum. As a result of that, the cells which is present in here encircle around this region. From the right side, this sclerotum and left side sclerotum, they coalesce and forms together a common mass with the scleral tissue which forms the vertebral body. So we have the vertebra, vertebral body is there. So this particular vertebral body is formed from the sclerotum tissues. And this particular sclerotum tissue moves in different directions, like ventrally, laterally, moves on different directions and forms these projections on different directions, like transverse process, neural arch around this particular neural tube. And this is how the vertebra is formed around this particular neural tube, which is developing in region, which is the spinal cord. So this is how this particular region is formed. Now this particular vertebra, as you are seeing here, which is formed, the body of the vertebra, and this particular process coming on the lateral and this particular dorsal side, forming this particular neural arches and this particular transverse arches, costal element, which is coming here in forms of the rib, they comes from this particular sclerotum tissues. Now, in between vertebra, we have a disc. We call them as intervertebral disc. We refer that intervertebral disc. It has two components, the central component and this particular outer component. The central component, we call them as nucleus pulposus. That nucleus pulposus, which is present in the central part, is contributed only by the notochord. That notochord around which this particular entire thing is formed. And surrounding this fibrous annulus fibrosus is contributed by that particular scleral tissue or sclerotum tissues. So this is how these vertebrae or components of the vertebrae are formed around this particular spinal cord. So we have the vertebral body, we have the transverse process, we have this neural arch that is contributed by the scleral tissues, which is known as sclerotum tissue, which is paraxial mesoderm, and we have this intervertebral disc. It has a two source, the central part and the peripheral part. Central part is that we call them as notochord tissue that contributing it, and rest of this particular notochord gets absorbed, and surrounding this nucleus pulposus we have an annular fibrosis. That particular element is making this outer rim of this particular intervertebral disc. So notochord should normally, in a normal healthy individual, should be present as the nucleus pulposus. That's the only legit existence of notochord in adult human body. Rest of the notochord should be replaced, absorbed, destroyed, etc., etc. If any remnant of the notochord remains to be there beyond this particular nucleus pulposus, that remains as an abnormal tissues, and then we call this abnormal tissue growth over here as chordoma, which may be most commonly we can see this chordoma present around this particular cervical region or lumbosacral region where it develops into. So this particular chordoma is a notochordal tumor or notochord remnant tumor, which normally should not be present. So this is what transforms from the somite. What is the somite? That is the paraxial mesoderm. The paraxial mesoderm transforms into sclerotum. Sclerotum forms what we have learned. It forms the myotome that gives rise to the muscle. It forms the dermatome that forms the skin. So this is what develops entirely from the somite. Somite means paraxial mesoderm. Next to paraxial mesoderm, we have the intermediate mesoderm. From the intermediate mesoderm, that tiny tissue develops from the genitourinary system. That means it develops the kidney from there. It develops the gonads from these particular tissues, which is intermediate mesoderm is. So this particular intermediate mesoderm transforms into this particular element. Then we are left with the lateral plate mesoderm. We have talked about lateral plate mesoderm beforehand as well. So this particular lateral plate mesoderm holds a body cavity which swings around this particular amnion and yolk sac, and this particular cavity which holds here, which is going to be the future, the intra-abdominal body cavity or intrathoracic body cavity. So that particular element swings around, forms this particular element. We call them as the intra-embryonic coelom or intra-embryonic body cavity. So this particular layer, parietal layer, visceral layer, is forming around this particular central body cavity that we have, forms this particular lateral, comes from this particular lateral plate mesoderm. So this particular fate of individual mesoderm is very important. We see that this particular thing is several times coming as an exam. as fate of this particular each component of the mesoderm, like a somite component, like intermediate mesoderm, and this particular lateral pin mesoderm. So this is one of the question that is there in the last year. So we'll see that how it is coming in the further year as well. So this is very important region that you must prepare about this element. So this is the first section of this discussion today that we are going to have. Transformations of these different mesoderms into different components. So then we are left with this embryo to talk about what further development happens to the embryo beyond that part. But before that, let's see what you are doing at home. I understand that you are not studying, but that's your choice. If you're not studying, you can expect you are not passing. That's guaranteed. So anyways, so I will flash some questions in here. Try to answer them how they are coming. Which answer? Okay. This brings us to the end of this particular session on embryo. So we have this embryo developed this far, we call them as disc. Now we are cylindrical as you consider ourselves as an organ. So from this disc, which is a leaf shaped structure, we have to transform into tubes. So we have to just like do some maneuver so that that particular leafy element can be transformed into a tube structure. So this brings us this particular phenomenon, which is one of the remarkable thing that happens to this embryonic development. We call them as folding. So this particular part of this particular embryo go on bending. So we call them as the folding of the cranial part or bending of the cranial part, bending on the caudal part and bending from the lateral part. So this particular embryo, which was a disc beforehand, has to go like this, bend it and come close and form a tube. This particular cranial part and the caudal part also has to bend to accommodate the differential growth of different organs. So we have developed this particular neural tube so far. The neural tube going to go to form this particular brain, and the brain growth in this embryonic life grows in a very exponential growth. It's a very heavy growing tissues which is going on, because you can understand this particular human brain, which is the only thing standing against or standing between anything else other than this particular rest of this animal species. So this particular brain has to grow really aggressively so that this human species can have this particular forebrain, hindbrain and midbrain can be formed. So as a result of that differential growth on the differential tissues, this particular embryo keeps or shows some kind of flexure or bending. That particular bending we call them as folding, which is this particular germ disc getting into folds over there. So there are three forms of bend that is observed in this particular embryonic phase. A bend which is happening in the cranial part, a bend is happening in the caudal part, which is forming in these two axes, and then bend we have on the lateral extremities. This is going on. So we have a link on this YouTube. If I have time, I can just like display as well. You can just like go and check with this YouTube displays, which gives a visual about the animations of this particular folding. Now this is what we have developed so far. We have this particular tube. Now we have a sequence of the thing. If you are looking from the top, we have this particular tissue, which is cranial most part of this embryo is lying. This is the cranial most part, this is the caudal most part, it is connected with a connecting stalk to this particular region. Now you see that most anterior tissue which this particular embryo has developed over here, we call them a septum transversum. This is a connective tissue which is formed in this particular cranial most part of this particular developing embryonic disc. This septum transversum eventually in an adult develops into a part of it develops into that the diaphragm, which we just addressed in this previous day session. So the septum transversum, which is most cranial most. Then we have this particular circular element, we call them as cardiac or pericardial sac, which is also starting to grow in this particular during this particular 4th week of life. So this particular embryonic region, which is next to this particular septum transversum, is this particular cardiac cavity, also is known as pericardial cavity, with this developing cardiac sac. And then we have this particular region, we call them as buccopharyngeal membrane, which we mentioned about the 2D region, 2-dimensional. That means it's the only region where we have these two layers existing, which is the epiblast and the hypoblast only existing in this layer. And then we have this particular neural tube, notochord and everything together going on. So this is the front to back, this was this element which is there. Now this is what we are seeing here, the neural tube is growing, and this particular neural tube keeps on growing and overlooms over this particular growing tissue and grows over there. If you see this particular element, this particular neural tissues comes on over here, which is going and further developing. So as this particular neural tissue develops, as a results in the cranial most part loops back or makes a bend like this. So this particular bend, which is created in this because of the overgrowing of this particular neural tissue along with this particular thing, pushes this particular structure down and ventrally. So as a result of that, this particular embryo, this particular brain grows, and this particular growing brain grows further and pushes this content which was anterior to it down and under. That means go anteriorly and goes in ventral aspects. So as a result of this, the orientations of this particular structure that we have seen, septum transversum, cardiac tissues, and then we have seen this particular buccopharyngeal membrane switch the position. So this is before this particular bending happened. We have the sequel of the thing, septum transversum first, then we have the cardiac tissues, then we have the buccopharyngeal membrane, oropharyngeal membrane, then we have the forebrain. Now forebrain overgrows over these regions and grows above and grows heavily. As a result of that, the weight of the growing vein pushes this particular cardiac tissue down, pushes the septum transversum down, pushes this oropharyngeal or buccopharyngeal membrane down, and then it moves, curves into ventral aspects and results in the final positions of this. So it switches the position. So then we get this particular position. First is forebrain, then we get this particular buccopharyngeal membrane, then we have this heart, and most lastly we have this particular septum transversum pushed down and reaches to near to this particular throat region. So then we have further development of the brain, which is making this particular cranial thing goes farther and eventually pushing down to the septum transversum, reaching to this particular region. We call them as the thorax region, or the junctions of thorax and abdomen, and it stops there. And then we have this cardiac tissue lies on top of it, and then we have this mouth which is formed here. We call them as oropharyngeal membrane or buccopharyngeal membrane. Then we have this particular head, neck, and the brain developed. So this is how this particular cranial folding or head folding happens and switches the orientations of the structure. Initially this particular orientation was like this, and then because of the overgrowing brain pushes these things down, and this particular thing ventrally switches this particular septum transversum into this particular lower in position, ventral moves ventrally more, and then we have the cardiac tissue lies on top, and then we have this oral cavity, and then we have the brain. So this is how this particular orientations or switching of this particular thing happens. We call this particular bend as cephalic bend or head fold bend. So this head fold bend happens to occur in a cranial post part of this particular embryo. Then similar kind of bend or similar kind of change happens in the tail end of this embryo. So again the sequence matters here. So if you look at this particular sequence of the structures that is present in here, from to here, this is the primitive streak or primitive streak which is present in this region. This is before bending which is happening. So primitive streaks was there. Then we have this cloacal membrane. Then we have this allantois, which is out pouch of this yolk sac. So this is a sequence of anterior to posterior. This was there. Then we have this bend which is taking place because of this particular primitive strip disappears, and this allantois and everything goes down, and yolk sac moves inside. So result of this particular element, this is carving down and switches the position likewise. So as a result of this, we have the sequence before the bend or before the folding. We had this primitive streak, then we have the cloacal membrane, then we have the allantois, and as a result of that, when this particular bend happens in the tail end or tail region, the switch of position happens to occur. So this particular allantois moves in the front position. So this is the allantois which grows into this particular yolk connecting stalk. Then we have this particular cloacal membrane. Then we have this primitive streak happens in the back region, which forms the sacral region. Okay. So this particular bend or folding which happens in the caudal region, we call them as tail bend or caudal bend, which switches the position of this particular embryo in the lower pole and switches the positions of these embryo structures in the cranial region. So this particular bending which is happening to take place, this cranial fold and the caudal fold, they are happening simultaneously at the same point of time. It is not that like the head fold is happening and followed by the tail fold. They are happening to bend like in the same point of time because of the overgrowth or differential tissue growths. So this is carving down. This is the two thing which is making this particular thing, embryo, to carve. Now this particular curvature which is completed or accompanied by another fold which is known as lateral bend or lateral fold. So this particular fold, we call them as lateral fold. As a result of this particular amnion drops down or amniotic cavity drops down, and this particular yolk sac cavity sucks in. So you see that the portions of the yolk sac getting inside this particular intraembryonic coelom or intraembryonic body cavity and gets inside, and slowly this particular lateral extremities, right side, left side, folding and nearing together and closing the cavity and makes this particular cavity which is we are acquired with the body cavity. So this intra-abdominal or intrathoracic body cavity created by this particular dropping down of this particular lateral extremities of these folds and making this particular thing fuse together and forms this. Remarkable thing with this particular lateral fold over here, you see that this is a yolk sac. A portions of the yolk sac is taken by this particular developing embryonic fold, and a portion gets inside the body cavity and forms this particular tube. This particular tube, which is derived of this particular yolk sac, going to make the future gut or the gut tube. So all the gut tube that we have, the small intestine, large intestine, and the derivatives of the gut tube, that means this particular organs like liver, the pancreas, everything is developed from this particular gut tube. So this particular yolk sac tube contributes to this particular formations of the gut tube inside this intra-abdominal cavity or intracoelomic cavity. That intracoelomic cavity is formed by this lateral plate mesoderm, which is falling down and looping here and meeting in the midline and creating a cavity and fusing together and sealing this cavity. So this is how this particular thing is happening. So this particular entire thing of this particular embryonic development that happened during the time of 3rd week to 4th week, and by the end of this 5th week, this particular entire phenomenon is, so we have this particular folding which is taking place in this different visuals. It is making cross sections. We have the trilaminar layers which you're seeing here, this yolk sac and the umbilical cavity which you're seeing here, taken over here, cross section coming over here, and we have the 3 layers. And this particular 3 layers in 3 different regions, we will be seeing this particular folding to be taking place. So we have this notochord, middle portions, and 3 layers is formed. We have this particular lateral plate which is creating over here. We have this particular transformations of this middle part of here. We call them as paraxial, intermediate, and the lateral plate mesoderm. We have the cavity formed inside to it, and this particular thing shrinks over here. So we have these particular things ready for the folding now. Now this element which you are seeing here is ready for folding, and this is a folding which is in the lateral extremity you are seeing here. The intraembryonic body cavity which is taking a part of this particular yolk sac, which is getting inside and sealing over here and creating the gut tube like this. So this is how this particular lateral folding which is taking place in this particular transverse plane. We have another sections which is in the higher division, which also figures out how this particular lateral folding which is created in this particular cranial most part. So this is a caudal region which see that this particular lateral folding which was helping it out, and this is a little bit in a cranial position, this folding over there. So same transformations is going to take place, neural tube formations, this particular the paraxial mesoderm transformations, and this particular gut tube is getting inside, and the head fold and tail fold and lateral folding which is taking a part of the yolk sac in here, and this fusing this part of the thing and creating this particular intraembryonic body cavity which holds this particular yolk sac derivative as the gut tube and which hangs by the mesentery. In a sagittal section, if you see that, you can see this particular cranial fold and the lateral fold which is a tail fold which you can appreciate. This is the cranial end, this is the caudal end, your connecting stalk, and this is the allantois in the cranial end, and this is what we see that heart and the septum transversum has, and this is the brain tissue where it should be growing in here. So this is how this particular folding over there, neural tissues which is coming over here and making this particular growing farther bulging and resulting in this particular tissue moving in, and as a result of this thing is switching the position and holding this particular region. So we see the septum transversum, cardiac tissue, buccopharyngeal membrane which is placing in this particular region, and the brain tissue which is coming in here. Similar to that, we have the allantois which is placed in the front, then we have this particular region, and then we have this primitive state. So this is how this particular entire thing of the folding happens to occur. You can see several animations are there which helps you out to further understand this particular process. We do not see this particular folding coming as a exam short note or any kind of forms, but this understanding is important for us to understand this thing happened, and this particular thing is the only thing that is transforming this particular transforming us into the tube-like organ or tube-like structures. So we have to know about the gastrulations, we have to know about the neurulations, we have to know about these transformations of these tubes. Slowly, when you go on the systemic embryology, we will talk about the cardiac tissue or cardiac cushion formation that you are seeing here, the cardiac tube, how it is forming, and we will be talking in detail about it. We will be talking about the brain formations, we will talk about the ear and the eye formations in the subsequent, which taking place between this 5th to 8th week of time, which is going to form. We see this particular pharyngeal arch, we will be talking about the development of the pharyngeal arch, how it is forming to form the neck region, how this particular pharyngeal arch contributes to form the blood vessels, etc., etc. And we'll be seeing this particular from this tube that you have formed after the folding. The buds are coming in the upper extremity and the lower extremity. We call them the upper limb bud and the lower limb bud. They are going to form these arms, legs for us. So these are the way this is going to. So this is early 5th week picture. We see that the embryo is entirely folded, the brain is nearly formed, and this particular thing which is coming over here, the limb buds, the upper limb bud, lower limb bud, and this particular somites is disappearing from this particular paraxial mesoderm, slowly disappearing. This is the connecting stalk which is connecting over here. Gut is formed, the heart is formed over this region. This is the stomach region which is going to abdominal region. This is the tail region which is going to form in this aspects. So this is the entire 5th week, and for the development, this is, you can see this depressions over there. This depression is going to form these ear placodes. The ears will be coming from this region, and from the frontal region the eye placode will be there. So eye will be developed from this particular end. So we'll be just like developing and talking further. So this covers this particular entire thing about this particular embryonic development, starting from this one cell stage to this particular multi-stage cell. We'll talk about the embryo later on. Now, before we move to the embryo further, we have to finish this particular placental membrane. So we'll talk about the placental membrane next day, and clinical related thing which is required for us for these undergraduate exams in the next subsequent class, and we'll be just like log off from this session till the exam begins. After the exam, we'll be starting with the systemic embryology. So that's the plan. Study well. Don't expect to be getting passed without studying, without scoring, without writing, without answering. Thank you.
Cecum. First part is the cecum. Then next is the appendix. Don't forget the appendix. Okay. Then next one is ascending colon. Then transverse colon. Then descending colon. Sigmoid colon. Then rectum, anal canal. Okay. So write it down. The large intestine consists of cecum, appendix, ascending colon, transverse colon, descending colon, sigmoid colon, rectum. Okay, sigmoid colon and next part ki rectum. Okay, then the last part is anal canal. Okay, so this is about the large intestine, right? But what is the peculiar thing here? If you say the transverse colon, how does it is belong in same level? No, তাই তো দুটো কি সেম লেভেলে থাকে? Transverse colon is a horizontal colon, right? So is it become in same level for right side, left side? No. Which side is below? Right side. কেন right side below হবে? Because এখানে কি আছে? Liver. ঠিক আছে? So liver is occupied this portion. Appendix পাওয়া গেছে। আর কিছু নেই? Simple columnar columnar. Okay. So what I am talking about the transverse colon. Okay. So right side is little bit below. Okay. Why? Because the presence of liver. এটাকে কি বলে? রাইট সাইডের কর্নারটাকে বলে হেপাটিক ফ্লেক্সার। ওকে, রাইট ইট ডাউন। দ্য রাইট সাইড অ্যাঙ্গুলেশন অফ ট্রান্সভার্স কোলন, দ্য রাইট সাইড অ্যাঙ্গুলেশন অফ ট্রান্সভার্স কোলন is also known as hepatic flexure. It is also known as hepatic flexure. Okay, and hepatic মানে কি liver? Okay, hepatic flexure, 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 hepatic flexure. And left side angulation of transverse colon is known as splenic flexure. Okay, left side angulation of transverse colon is known as splenic flexure. ঠিক আছে, অল্টারনেটিভ নাম কি আছে? হেপাটিক ফ্লেক্সারের অল্টারনেটিভ নাম কি হবে? রাইট কোলিক ফ্লেক্সার। ওকে, সো ওয়্যার ইউ হ্যাভ রিটেন দ্য হেপাটিক ফ্লেক্সার, রাইট ইট ডাউন, রাইট কোলিক ফ্লেক্সার, রাইট কোলিক ফ্লেক্সার। Okay, and left side splenic flexure টাকে বলে left colic flexure, left colic flexure. Okay, এখানে just put a star mark. The splenic flexure is 2.5 centimeter higher than the hepatic flexure. So got my point? Splenic flexure is above, hepatic flexure is below. Okay, so we will go for the diagram, then we could understand. So what are the function of large intestine? So absorption of water, which is very much important. Okay, just ask some patient to take the water, you put the stethoscope, okay, in the abdomen. So you will find there one is a gargling sound where at the point of large intestine. বোঝা গেল? কারণ জলটা যেটা খায় সেটা পুরোপুরি স্টমাকের যে পাইরোলিক ক্যানেল দিয়ে পুরো চলে যায় কোথায়? Where it will go? It will go to the large intestine. Okay, so water absorption mainly taking place where? To the large intestine. Okay, and rather than which are the other subject? Other object that would be absorbed? Drugs. Very good. Drugs are minerals, electrolytes. Okay, electrolytes. Okay, are ke absorb hoy? These two things, one is vitamin B12 that you all know, vitamin B12, arekta ki vitamin K. Okay, so as because these two, you know, the vitamin B12 and vitamin K is very much essential for what? Coagulation. Already I told you which is the vitamin K dependent pathway. Coagulation pathway পড়েছিস physiology তে? Okay, so vitamin K dependent pathway is কত 2, 7, 9, 10. Write it down. Vitamin K dependent coagulation pathway is 2, 7, 9, 10. 2, 7, 9, 10. Okay, arekta ki hematopoiesis. Hematopoiesis is very much important thing. Okay, so these two vitamins are very much essential. Suppose anybody is not taking the non-vegetarian food. Okay, so what would be the deficit factor? Vitamin B12. Okay, and vitamin B12 is very much essential for nerve. ঠিক আছে, so you will find who are vegetarian, they chronically they are having some kind of neuropathy. ঠিক আছে, বুঝতে পারলি? তাহলে নন ভেজ খেতে হবে। ঠিক আছে, okay, large intestine. So this is the main function. So see, this is what? This is the cecum, this is the appendix, this is the ascending colon, this is the transverse colon. So এটাকে কি বলবো আমি? হেপাটিক ফ্লেক্সর। কেন হেপাটিক ফ্লেক্সরটা নিচে? কারণ it is right side occupied by liver. Okay, and left side this is the splenic flexure. Okay, so this is higher, কতটা higher? 2.5 centimeter. And which thing is passing through this line? That is known as transpyloric plane. মনে থাকবে? Which one is crossing this transverse colon? সেটা হচ্ছে transpyloric plane. Transpyloric মানে কি? It is traversing to the pylorus of stomach. ঠিক আছে, সারফেস অ্যানাটমিতে এটা ভীষণ ইম্পর্টেন্ট। ট্রান্সপাইলোরিক প্লেন, ঠিক আছে, এই প্লেনটাকে বলছে ট্রান্সপাইলোরিক প্লেন। Then next one is descending colon. This is the sigmoid colon. Then rectum and anal canal. Okay, so this is the part of large intestine. So rather than absorption of water, formation of fecal mass and production of mucus. Mucus is also very much important. You will get lots of goblet cell over there. Okay, in the large intestine. Okay, so see, this is the circulature. Ever if you see the gross anatomy, okay, gross anatomy of large intestine. Gross anatomy means macroscopic anatomy. You could see by your eyes, right? So what are the three features? Write it down. Three feature, macroscopic feature of large intestine are, এবার ভালো করে দেখ, ঠিক আছে, লার্জ ইন্টেস্টাইনটা ভালো করে দেখ। What are the characteristic? First one is you are seeing lots of this kind of thing. These are known as hostration or sacculation. ঠিক আছে, so number one feature কি লিখবি? hostration. দেখ, just see the spelling over here, hostrum. Okay, so hostration হচ্ছে first feature, hostration or sacculation. Okay, then second feature কি? If you see that throughout the large intestine, you could see at the middle there is one band like structure. Okay, You could see it is coming throughout the gut, this middle. A band-like structure, this is known as tinea coli. These are known as tinea coli. So second one is tinea coli. Okay. So first one, hostration or sacculation. Second one is tinea coli. A third ki pabo, if you see this band-like structure, you could see there are some fibrofatty tissue that are coming out from the tinea. What it is called? It is called as appendices epiploicae. এখানে কোথাও লেখা নেই. Okay, just write it down. Appendices epiploicae. Appendices epiploicae. So these are the three features. Appendices epiploicae. EPILOCIAE. Epiploicae. Okay. Appendices epiploicae, if you see, there are three types. Okay, so we will study in gross anatomy যখন আমাদের abdomen হবে তখন. So just for now, just remember three features. একটা কি hostration or sacculation, then tinea coli, then third one is appendices epiploicae. Okay, so if you see the appendix, এখানে কি tinea দেখতে পাচ্ছিস? So there is no tinea, no hostration, no appendices epiploicae. একটাও feature থাকবে না. Okay. And if you see the rectum, which one is absent? Tinea coli nahi, appendices epiploicae nahi. Okay, but hostration is present. Okay, so by that way you could say the features. So see, this is the abdominal aorta and how the blood is going throughout the large intestine. Okay, so what it is called? It is called as mesentery. Okay, it is called as mesentery. Now this thing already we told how the feces is formed. These are formed by the indigestible food particles along with water and electrolyte. Okay, and it lacks intestinal villi. Key main feature. So have you studied small intestine? No, not yet, right? Histology hoyeche small intestine? Hoyeche? So you know what are the features of small intestine? কোথায় হলো? স্টমাক হয়েছে তো স্যার পড়িয়েছেন। আচ্ছা, তাহলে কি কি ফিচার আছে বল স্মল ইন্টেস্টাইন? মাইক্রোভিলাই, ভিলাই এন্ড মাইক্রোভিলাই। আর কি থাকবে? পেয়াস প্যাচেস। আর কি থাকবে? ব্রুনাস গ্ল্যান্ড। ওকে, so these are the features of small intestine, but you would not say any intestinal villi. Okay, but what is the differentiation? So write it down. These are the features for vermiform appendix. Okay, histological feature. So you have seen appendix is a small structure, minute structure, so lumen would be small. Number one is there would be small angular lumen compared to the thick wall. Second one is there is no villi. There is no villi. Third one is there is no crypts. Okay, so throughout all the histological specimen you will get three which are having star shaped lumen. Kar kar thakbe? Ureter. Okay, second one is appendix, third one is vas deferens. Okay, so these three you will get small lumen afterwards throughout the structure. Okay, because these three are like the pipe. Okay, so these three you will get. So what is the basic characteristic histological feature? You have to write, that is ring of lymphoid follicles. Appendix ke ki bole? Aggregation of lymphoid follicles. Okay, tonsil ke ki bole? Aggregation of lymphoid follicles. Suppose any infection in the stomach, first thing to inflame ke? Appendix. Okay, so that's why ring of lymphoid follicles with germinal center. So what is germinal center? লিম গ্ল্যান্ডে অলরেডি আমরা পড়েছি কি জার্মিনাল সেন্টারটা। সো আউটসাইড দেয়ার উড বি ওয়ান হ্যালো। ওকে, সো দ্যাট ইজ নোন অ্যাজ জার্মিনাল সেন্টার। ওকে, অ্যান্ড where it would be present at the lamina propria. So you know mucosa has got three layer. কি কি লেয়ার বলেছিলাম? epithelium, then lamina propria, then muscularis mucosa, right? So lamina propria has got these lymphoid follicles. Okay, so write it down, ring shaped lymphoid follicles consisting of germinal center at the lamina propria of the lumen. At the lamina propria of the lumen. Okay. Then what about the muscularis mucosa? Muscularis mucosa is not adherently formed, it is disrupted. Okay, this is another characteristic, disrupted muscularis mucosa. Disrupted muscularis mucosa. Okay, so these are the features of vermiform appendix. Okay, so see, this is the picture. So what are the thing you could see? The lumen is small and it is irregular. Okay, where you have written the small, there you write it is irregular also. Okay, it is not circular, you could see. Okay, so first layer আমরা কি পাবো? Epithelium. So epithelium contain very few intestinal gland. Okay, very few intestinal gland. Okay, and epithelium consists of so many cells. দেখ কতগুলো cells আছে? First one কি থাকবে? Columnar cell, goblet cell, Paneth cell. Paneth cell কি secrete করে? Lysozyme secrete করে. Enteroendocrine cell, then M cell, then lamina propria. Okay, so write down these are the structure, these are the cells: columnar cell, goblet cell, Paneth cell, enteroendocrine cell and M cells. Okay, lamina propria we already told কি কি পাবো? The lymphoid follicles. কোনগুলো lymphoid follicles? You could see this violet color circular thing. Okay, so these are the lymphoid follicle. কোথায় থাকছে এটা? Lamina propria. Okay, and if you see the muscularis mucosa, see it is disrupted. You could see here it is not there. Okay, again here it is not there. Okay, so disrupted muscularis mucosa. Is it? Muscularis mucosa disrupted. Okay, then submucosa te ki thakbe? Lymphatic tissue. Next layer is submucosa. Dekh etate maje maje in between you will get what? These are the lymphatic tissue. Okay, and last one is muscularis externa. You know, two layers: inner circular, outer longitudinal, and the last one is serosa. Okay, so you have to say this much about the appendix. Okay, first epithelium, first lumen, irregular lumen. Epithelium, how many cells are there? Okay, then next one is lamina propria. You have to say the lymphatic follicles having germinal center. Okay, then muscularis mucosa is disrupted. Submucosa, few lymphatic follicles are there. Then coming the muscularis externa having two layer: inner circular, outer longitudinal. Last one is the serosa. Okay, so one by one you have to explain. This is the appendix. Now about the cecum and colon. Okay, so what are the histological feature you will get? First one is no villi. So whenever you are reading large intestine, you have to write there is no villi. Number one. Next, crypts are well developed and lined by plenty of goblet cell. So ekhane each and every intestinal crypts are very well developed. Okay, and they are lined by goblet cell. This is the second feature. Okay, then Paneth cell are absent on the crypt. See, cecum and colon does not have Paneth cell. Paneth cell kothay ache? Appendix e peyechi. Why? Because it has to destroy the different type of microorganism. Okay, so that's why Paneth cell is present on the appendix. Okay, then outer longitudinal layer of muscularis externa shows thickening of the ribbon like bands. This thing I told, ribbon কতগুলো যাচ্ছে? তিনটে. There are 3 layer of ribbons that is running throughout the colon. These are known as tinea coli. Okay, so write down this feature. Outer longitudinal layer, কোন layer এ থাকে? That is also important. Outer longitudinal এ থাকবে. Okay, outer longitudinal layer of muscularis externa having thickening form of ribbon. Thickening form of ribbon like the bands in 3. These are known as tinea coli. These are known as tinea coli. Okay, and serosa te ki থাকবে? That, see, this is the spelling of appendices epiploicae. Okay, so what is this? These are the fat filled pockets. These are the fat filled pockets. Okay, so serosa shows fat filled potential pockets. So these are known as appendices epiploicae. Okay, so these are the all feature you have to say about the cecum and colon. Okay, so lumen ta kemon hobe? How about the lumen? Lumen would be larger and it would be circular, irregular hobe na, regular lumen hobe. Okay, so we are not having the picture. So see, this is the large intestine. Okay, so large intestine cross section ta kemon bhabe neya hobe? Just longitudinally, right? So what is this layer? This is the epithelium, right? So in the epithelium consists of key type of epithelium thakbe? Simple columnar. Okay, and rather than that you could find there is no villi or plica circularis. Okay, mucosa te ki thakbe? Epithelium consists of, there is specific cell that is known as colonocytes. Okay, there are one type of specific cell that is known as colonocytes. Okay, and lamina propria, again it is having lymphatic tissue, lymphocyte and collagen. So these all are same. Okay, just where you have to mention the outer longitudinal layer, tinea colia. Okay, and this serosa you have to mention what? Appendices epiploicae. Okay, this thing already we discussed. All same thing are same like the GIT, general thing. Okay, crypt. So which are the crypt? So see, they have shown one microscopic structure here. This is this layer. Okay, so in this layer what you could see, these are the crypts. Okay, these are the crypts. A crypt se ki thake, which is present over there? Intestinal gland. Intestinal gland will secrete what? Mucus. Okay, so this is all about the colon. Okay, then what is this layer? They have shown next layer. This is the first layer, this is the epithelium, then this is the lamina propria, right? So this is the lamina propria. So you could also get some kind of lymphatic tissue over there. Okay, so all the waste material, right, they are going over there. So it may be some kind of poisonous thing to our body. So body has to give a fight. So this is nothing but the lymphatic follicles. Okay, next coming to the rectum. So before going to the rectum is misnomer. ঠিক আছে, rectum নামটা দেওয়াটা ভুল। rectum মানে কি? Where else you got the rectum word? Have you got ভাষা rectum? কোথায় পড়েছিস? Where you got? Very good, nephron এ থাকে। কোথায়? juxtaglomerular apparatus এ পেয়েছিস। সো রেকটাম মানে কি? স্ট্রেট। ঠিক আছে, কিন্তু এখানে রেকটাম কি? ইট ইজ নট অ্যাট অল স্ট্রেট। ওকে, ইট ইজ মিসনোমার। অলওয়েজ রিমেম্বার ইট ইজ মিসনোমার। ওকে, সো রেকটামে হিস্টোলজিক্যাল ফিচার কি পাবো? লং ক্রিপ্ট অফ লিভারকুল। সো দিজ আর দ্য ইন্টেস্টিনাল গ্ল্যান্ড। এখানে ক্রিপ্টটা কি? অনেক বেশি লং। Okay, so there is long intestinal crypt, so these are known as crypt of Lieberkühn. এটা দিতেই হবে ফিচারে রেকটামে। Okay, then next one is lymphoid tissue is less abundant in the lamina propria. Lamina propria তে lymphatic follicles থাকে না। Okay, less abundant. Then the muscle coat is lacking, that is tinea coli. You have already shown tinea coli থাকে? No, কি থাকে? Hostration থাকে। Okay, somewhat hostration or sacculation, and serosa replaced by adventitia at the lower part. Rectum in lower part act as a blood vessels, like a blood vessels. ঠিক আছে, what are the three layer of blood vessels? তিনটে layer কি কি? Inside to outside is IMA. Okay, so first one is intima, then media, then the adventitia. So adventitia is the last layer, and which one is elastic? মাসেলটা কোথায় থাকে? স্মুথ মাসেল থাকে, তো ব্লাড ভেসেল যে ভ্যাসোডাইলেশন ভ্যাসোকনস্ট্রিকশন করে, সো ইট ইজ প্রেজেন্ট অন দ্য টিউনিকা মিডিয়া। তাহলে অ্যাডভান্টেশিয়া কনসিস্ট অফ কি? ধর ইন্টিমা হ্যাভিং দ্য এপিথেলিয়াম, রাইট? এপিথেলিয়াম ইনসাইডে আছে, তারপরে হচ্ছে মিডিয়াতে হচ্ছে মাসেল লেয়ার। হোয়াট অ্যাবাউট দ্য অ্যাডভান্টেশিয়া? It is type of connective tissue, nothing else the connective tissue. Okay, so a lower layer of the rectum is converted to the adventitia. Serocytes not present, only on the lower layer. Rectum is divided into two parts, upper part and the lower part. Okay, so these are the histological feature of rectum. So see, this is the rectum. So how does it look like? Already I told you, it is not at all straight. Okay, it is misnomer, and this is what? This is known as rectal valve. আমরা যখন gross anatomy তে যাব, again we will study each and every part, okay, of the GI tract. So now see the histological feature. What are the thing you will get? Goblet cells are abundant. Okay, glands are deeper. So you could see the glands. So what is the difference between the rectum and the colon? যদি histological differentiation করা যায়, what is the difference? Crypts থেকে শুরু করতে হবে. Crypts are smaller in size in where? Large intestine, but it is long in the rectum. Okay, then second point কি হবে? Glands are present deeper where? In the rectum. Okay, but it is present superficial on the large intestine. Okay, and then you could see this complete muscle coat. Rectum is very strong structure. Okay, so that's why this muscle coat, see how nicely it is formed. Okay, so there would be complete muscle coat. Okay, and remaining whatever you studied, it would be same like the GIT. Okay, you may include the lower part of rectum, last layer serosa is converted to adventitia. You may write. Okay, so these are the feature of rectum. Rectum slide is not important for your MBBS. Okay, so it is for MD. So you just study large intestine and appendix. Okay, so that would be enough. So this thing already we told. This thing you wrote. So epithelium of the appendix ki hobe? Simple columnar epithelium, and there would be few crypts. These are known as crypt of Lieberkühn. Okay, lymphatic nodules kothay thake? Lamina propria, small angular lumen compared to the thick wall, absence of intestinal villi. Okay, so this thing already we discussed. So see, this is the picture. So first one epithelium, at first irregular lumen, then this is simple columnar epithelium. Okay, next one is in the lamina propria you are getting this circular thing, these are the lymphoid nodules. Then disrupted muscularis mucosa, it is not having the circular wall. Okay, it is irregular disrupted muscularis mucosa. Then in the submucosa again you are getting, see these violet colored dots. Okay, so these are what? These are the lymphatic follicles. It is still present on the submucosa. Okay, and the last layer is the muscularis external. Okay, so this is the appendix. We have got the appendix slide. Okay, this is also the appendix. Same thing. Okay, so if you see the lymphatic follicles, you could find here it is violet color. It is stained in violet. Okay, so see, this is the large intestine. If we are having the slide, how does it look like? So first one is, this whole is the mucosa. So you could see the crypts are small. Okay, but the gland is more larger. You have seen so many cells are there. Ki type of cell thake? Goblet cell, then Paneth cell, then 5 types of cells are na, then enteroendocrine cells, M cells are columnar cells. Okay, columnar cells is so many cells are present over there. Okay, and this is the muscularis mucosa. So you could find very nice lining. It is not at all disrupted. Okay, and few lymphatic follicles. This is present on the submucosa. Tell me the plexus I told. Submucosa te ki plexus thake? মানে মাসকুলারিস মিউকোসাতে কি প্লেক্সাস থাকে? মেসেন্টেরিক প্লেক্সাস সাবমিউকোসাতে থাকে, রাইট? সো হোয়াট অ্যাবাউট দিস টু মাসল লেয়ার? মায়েন্টারিক অর আয়ুর ব্যাচ, বোথ আর সেম, মায়েন্টারিক অর আয়ুর ব্যাচ। আর মেসেন্টারিকটা কোথায় থাকবে? সাবমিউকোসা, ইন বিটুইন দ্য মাসকুলারিস মিউকোসা এন্ড সাবমিউকোসা। ওকে, দুটো লেয়ারের মাঝে। আর এই দুটো লাস্ট লেয়ারের মাঝে কি থাকবে? আয়ুর ব্যাচ অর মায়েন্টারিক প্লেক্সাস, সেম প্লেক্সাস অফ নার্ভ and blood vessels. Okay, so see, this is the columnar absorptive cell. So this is, you could see, this is simple columnar epithelium. Okay, so you just take a snapshot. This is the snapshot for large intestine. Okay, this is the snapshot for large intestine. So this is the lumen. So just think about that. It is the large intestine. They have taken one slice. Okay, so that this structure and these structures are similar. Okay, both side you are having this longitudinal gland. Okay, and inside this one, in the submucosa, you are having these lymphatic follicles. Okay, so this is the slide for large intestine. Okay, so just go and see the appendix and the large intestine. Okay, so that's all.